Hi, my name is Jana Crawford. The date is August 10th, 2026, and this is my Unit 3 video presentation. This presentation was pretty much revolving around the question of non-coding DNA and the mystery behind that, and that's exactly what the author was kind of answering. And so the first question is, what is the non-coding DNA mystery? And so the non-coding DNA mystery was when scientists originally focused on genes that only coded for proteins, and so since a relatively small amount of uh, human genome directly codes for proteins, it begged the question of what all this DNA is doing if most of our DNA doesn't code for proteins. And so for a long time, these non-coding DNA was just labeled as junk DNA, which gave the impression that they were useless or had little to no purpose. So the author, Bernardi, argues that this is an oversimplification of what it actually is, and that his idea was that non-coding DNA can help um, have an important structural role, and it can help determine how DNA is packaged, how chromatin is organized, which regions interact with each other in DNA, and how chromosomes are arranged within the nucleus. And so the second question was Bernardi's main evidence for this. And so his main evidence for this was isocharies. And isocharies, if that's how you pronounce it, I have no idea, are large regions of DNA with relatively high consistent levels of guanine and cytosine content. And so Bernardi connects different guanine and cytosine compositions with different types of genomic regions. And so there's guanine and cytosine poor regions, and then guanine and cytosine rich regions. The poor regions had fewer genes, and the rich regions had more genes. Um, and so the next question was, how does DNA affect chromatin? Bernardi's important argument is that the DNA sequence itself can influence chromatin structure, and that DNA isn't just a passive set of instructions. It's a little more complex than that. And so its sequence and composition can influence where the nucleosomes are positioned, how DNA folds, how regions of DNA interact, and then how chromosomes are organized. And then um, his conclusion to that was pretty much that he proposed the idea of the genomic code, and that this is different from the traditional genetic code, um, and that the genomic code described by the author provides information that can help determine how DNA is physically organized. And so the order would be DNA sequence, then chromatin organization, then 3D genome structure. And this provided a possible explanation as to why there's so much DNA directly that doesn't directly code for proteins, and that non-coding DNA can still carry information for genome organization and chromatin structure. And in my opinion, for this article, I thought it was really interesting. I liked, seeing the different answers to the non-coding DNA mystery. I liked the author's answer and his exploration of it, I guess. And I think it really does transform the way we think about non-coding DNA. So before learning about this topic, I thought that if DNA doesn't code for a protein that, I don't know, maybe it was junk DNA or something. I don't know. Maybe it just didn't do anything. But the author kind of at least showed and explained that just because it doesn't code for a protein doesn't mean it's just like floating around doing nothing. It's still like served a purpose. And so I found it most interesting that DNA can have information that affects structure and not just protein production. And that non-coding DNA can contribute to how chromosomes are organized, and that the physical organization of DNA can affect how the genome functions. And I really think this advances our understanding of DNA as a whole, genetics, and then how non-coding DNA isn't useless and how it does serve a big purpose.